Hi, back again.
Hello.
We'll wait like a couple of minutes before we'll start, and then I'll introduce you to the audience.
Okay, that sounds great.
Hi, Frank. How are you?
Hi, Katerina. Hi, Ryan.
Hello.
Okay. Yeah, we can slowly start and go ahead and introduce you, and I think people will still be coming. Um, slowly in. So, but yeah, let's not make the people that are already here make them wait. So, welcome to the Science Society Club, and we are very happy to introduce you to Dr. Ryan Field. He really was so kind to make time for presenting his really amazing work. um, so yeah, we are. Thank you so much for being here, and I wanted to give you some information about Dr. Ryan Field. He's the Chief Technological Officer at Kernel, and it is based out of Culver City. I'm sorry, California, United States, and uh, Ryan has did his bachelor degree in electrical engineering and physics in North Carolina State University, and his master's degree in electrical engineering at Columbia University, and his PhD in electrical engineering also at Columbia University. And yeah, a little bit about Kernel. um, it's a team of neuroscientists, physicists, engineers, programmers, and experiment and operation experts that are driven by the belief that exploring and quantifying the human mind is the most important and consequential opportunity of our time. And yeah, I really admire that spirit. And with that, welcome Ryan to our club, and thank you so much for taking the time.
Yes, thanks for the introduction and thank you for having me. Before I get started, I will point out if you looked at the paper that's linked here in the room, there are about, I think, 30 authors listed. So the work I'm presenting tonight is really a huge team effort from everyone who's been at Kernel for the last couple of years as we built this technology. And our team is just about 100 people now. So the company has grown quite a lot. So with that out of the way, I'll go ahead and just give you a brief outline of what I'll talk about for, I'll try and talk for about 20-25 minutes and then give some time for everyone to ask questions that you may have in response. So I'll start with just a little bit of background information about the technology and how it differs from some other types of brain-computer interfaces or BCIs. I will walk through some of the characterization results in the paper and explain a little bit about what they mean, highlight some of my favorite ones, and speak to the significance of each of them. And then at the end, I'll briefly talk about some of the results that we have from human measurements. So with that, I'll start just by setting the background here for the technology. There are a number of ways that you can measure signals from the brain. There are what I would call neuronal signals, or what are called neuronal signals, which are as close to direct measures of the electrical activity of the neurons in the brain as they fire. So if you think about groups of neurons, they're communicating through transfer of charge, which creates these electric currents and generate electric and magnetic fields. And so you can use techniques to measure the electric fields. This is commonly known as EEG, or you can use techniques to measure the magnetic fields. This is a technique called MEG. However, there's another indirect way of measuring brain activity, or when groups of neurons fire, and that's by looking at hemodynamic signals. And the kind of fundamental of how hemodynamic signals emerge is that when groups of neurons start to fire, they need oxygen to do the work that they're doing. And so your cardiovascular system provides oxygen local to the groups of neurons that are firing. So you get a fresh, kind of what I think of as fresh inrush of blood in the areas where your neurons are doing work. And activity is detecting changes in local blood oxygenation. And it's a technique that's commonly used with either fMRI or a technique called TD-fNIRS, or functional near-infrared spectroscopy. And what I'm going to talk about for the rest of tonight is fNIRS, which is this technique for using optics for measuring changes in blood oxygenation levels of the brain. So if you want a very common example of how this works, you can look at the latest watches from both Apple and Fitbit. They include a device, a sensor, that allows you to measure blood oxygenation at your wrist. So they use different wavelengths of light to measure the oxygen concentration of the blood at your wrist, and they give you some blood oxygenation percentage. Fundamentally, the technique that we're doing is very similar to that. So fNIRS generally uses two wavelengths of light, and these wavelengths are chosen specifically so that one is absorbed more by oxygenated hemoglobin, and the other is absorbed more by deoxygenated hemoglobin. And so as the local blood oxygenation changes, these two wavelengths of light, the two wavelengths of light, as measured by some detector nearby, will have their amplitude modulated. The intensity of the signal will change based on the local blood oxygenation. So this kind of absorption-based change in blood oxygenation has been around for decades. A lot of interesting work has been published on applications of fNIRS, so measuring the brain using this optical technique. And Kernel, as a company, kind of identified a way to take fNIRS and take it to the next level. And that method is called time domain fNIRS. So it's like using the traditional version of fNIRS, which is based on absorption, but now we're adding a time domain element to it, where we use very short pulses of laser light and measure the photons that propagate through the brain We measure how long it takes to get from where the laser fired to where the detector picks it up. And these types of systems have been developed in research settings, and the big advantage of them is that they provide more information than your traditional standard fNIRS systems. This additional information is that not only do you get changes in absorption, but you also get to measure changes in scattering of the tissue. And so time domain information adds a new dimension to doing fNIRS measurements. Further, because of the nature of this measurement, where you're pulsing a very short pulse of laser light and then detecting how long it takes photons to come out of the head near the detector, you get some information from this time of flight about how deep the photons went into the head. And this is really nice because this depth information will allow you to separate signals that happen in superficial layers, like our scalp, from those that are happening deeper from the brain. And the brain is really what we're interested in. The scalp is just a, if you think back to the Apple Watch example, the signal in the scalp will be very strong because you also have blood vessels in the scalp and your heart is beating, so you get this cardiac pulse. And so you want to be able to separate that strong scalp signal from the much weaker brain signal that happens underneath it. And so if I step back and talk a little bit about the hardware that's been used for these types of time domain or time of flight systems, they've always been done in kind of a rack mounted type of system. And there's this quote that's on the fNIRS Wikipedia page when it talks about time domain that I love. It's still there, and I don't want anyone from Kernel to go modify it. I want to see how long it stays up, but the quote is, "Time domain-based devices are totally immobile, space-consuming, the most difficult to make, costliest, hugest, and heaviest. Even so, they have the highest depth sensitivity and are capable of presenting the most accurate values of baseline hemoglobin concentration and oxygenation." So what we've done at Kernel is we basically canceled that entire quote. We've built Kernel Flow, which is a miniaturized and scaled version of the system that's designed to be wearable. It, in our opinion, is the best non-invasive technology that could be built in a way that would enable the broadest application of brain measurement to society. So we think it's a platform that you can build all kinds of brain measurement-based applications on top of. And kind of the favorite quote of mine from Brian, our CEO, is that what we've really done is the equivalent of making a mainframe into a PC. We've done that level of miniaturization, taking a whole room full of equipment and putting into a very small form factor that, for the first time, kind of brings in the possibility that it could be widely adopted and scaled. So with this background, now I want to talk a little bit about what we've done to demonstrate that the system we built actually performs the same or better than that rack-mounted hardware. Does our PC compete with the mainframe of TD-fNIRS devices? And this is what the JBO paper that we published really set out to show. So if you think about the brain and what it takes to measure it, and take one step back and say, how do you characterize a system that's meant to measure the brain, which is this biological system that's constantly changing, highly variable, and so hard to control? So what the researchers in the field have done is they've come up with these three standard protocols that they use to evaluate systems in the absence of a brain, so they can try and predict how well the system will work in measuring actual human brains. And so the three commonly used protocols are called the Basic Instrument Performance, or BIP protocol, the MEDFOT protocol, and the NeurOpt protocol. If you're interested in some of the work on these protocols, there was actually a collaboration that was based in Europe back in 2019 that looked at all the time domain systems in the world and characterize them against these three protocols. And that effort was called Bitmap. And I thought it was a really good overview of just where the field was in terms of the time domain systems. So in the JBO paper, we cover most of these three protocols, but not all. And I'll not go through every result in the paper, but we'll highlight some of the ones that I think are most interesting that show unique capabilities of our system performance. So the one I'll start with is what's called the responsivity metric. It's part of the BIP protocol, and the measure in the paper. But if you look at figure 11 in the paper, this is the result I'm going to talk about. So responsivity is really a measure of the sensitivity of our detector. And why this is interesting to me is that most time domain systems that have been built use these specialized detectors that are typically made from 3,5 materials. The reason they're made from these materials is because they're more sensitive to infrared light. Ours, on the other hand, is made from a standard CMOS or silicon process, and silicon is inherently bad at detecting infrared light. This is something we knew when we chose to design it in silicon, but we chose to build it in silicon because we wanted to build something that scales. It needed to have the same properties that all the consumer electronics that we buy have in order for us to scale it to something that could be used in the mainstream, and that's both low cost and widely accessible. And what we found through the characterization is that our detectors that we custom designed were able to maintain sensitivity that's in the order of the specialized detectors. And not only did we show that we could maintain the sensitivity of these detectors, we also demonstrated that we can record kind of record-breaking photon count rates with a constant responsivity. And so what I mean by this is if you look at the paper, we show the responsivity measure as a function of how many counts per second we pick up at the detector. And our detectors are capable of picking up over 1 billion counts per second. And if you look at most common, most of the rack-mounted systems, they're usually in the tens of millions counts per second. So we've really gone up by 2 orders of magnitude in the number of counts per second we're able to detect, and we've maintained the responsivity of the detectors. The next kind of result that I really want to highlight is what's called the instrument response function, or IRF. And this is what's shown in figure 12. And the most interesting characteristic of this, for me, is the dynamic range that you can see in this plot. So we're able to achieve almost 5 orders of dynamic range in this figure. And so what you see is the difference between the peak at 10 to the 0 counts versus the noise floor before the peak, which is just above 10 to the minus 5. So we've normalized everything here to a value of 1. So the peak is at 1 and everything else is normalized to that. So we have almost 5 orders of magnitude. And why this is significant is that the dynamic range allows us to use shorter channel lengths, or the distance between the laser source and the detector can be shorter, and we're still able to resolve changes in brain-related changes in the signal. And to me, this is important because it's one of the key advantages that allow you to build a compact and wearable system, which is being able to pack the sources and detectors closely together inside a module. But this is nice, it's a great result. And one of the questions is, can you have a wearable system that's also stable? Does it perform the same way over time? And so the next measure that I wanted to highlight was figure 13, which is a measure of the IRF as a function of time. And what you can see is that after a warm-up of about 20 minutes, the characteristics of that IRF curve remain pretty constant for the rest of the time. The thing that I am kind of proud of in seeing this is that unlike the rack-mounted equipment, where they may also have pretty stable IRFs over time, we don't use any temperature regulating devices to control either the temperature of the detector or the lasers. And again, this is an important consideration for building a wearable system. If you want to build something wearable, you can't spend the cost both in terms of weight and power of trying to cool off or maintain a constant temperature of the components in the system. And there are a lot more details on the BIP protocol that if you want to dig into, you can, but I'd like to move on to the next protocol, which is the NeurOpt protocol. And the main question that NeurOpt is trying to address is how well can a system resolve localized changes in absorption? So they have a couple of tests, and we did one of them where you use a cylinder of known properties. So it's made of PVC, it has a certain volume, dimensions are all specified by the protocol, and you place it in a liquid phantom. So this is a kind of a tank filled with a liquid that has known absorption and scattering properties. And those absorption and scattering properties are chosen such that they're representative of what you would find in the human head. And so in this test, you move the cylinder away from the source and detector. You move it away from the lasers and the detector and see how far you're able to resolve that change, that localized change in absorption due to the cylinder. And what we see in these curves, there are a couple of interesting things. So first, what you can see is that out past 20 millimeters, there is measurable contrast and CNR, which is a contrast to noise ratio, a measure of how confident we can be in resolving that contrast. In addition, each of the lines in this chart represent a different time delay after the laser was fired. So this highlights really what the power of the time domain technique is again. So if each of these windows is a 500 picosecond time window after the laser fires increasing. So window 0 is a time from 0 to 500 picoseconds. Window 1 is from 501 to 1000 picoseconds, and so on. And so what you can see here is that there's a function of how strong the signal is in each window based on how deep the cylinder is. And so this gets back to the beginning where I was describing how time domain gives you additional information based on traditional fNIRS systems. It gives you both scattering properties and it gives you an indication of depth based on the time of flight, how long it took the photon to come back out. So anyone who uses this type of system will find this type of plot really cool because it shows really that you can distinguish different depths of change in the absorption using the time domain information. So that's the one measurement from the NeurOpt protocol that we did. And I'll move on now to talk a little bit about the MedPhote protocol. So the MedPhote protocol is an array of solid phantoms, each with a known absorption and scattering property. So you have this two-dimensional array. I think there's a figure above in the paper that shows them with increasing absorption and scattering properties. And we choose the ranges of absorption and scattering to match what's expected again in the biological tissues we're trying to measure. And each of these phantoms is calibrated, and it's known to have these scattering and absorption properties. And so this is where we're going to try and flex and show that our time domain system not only improves on some of the things that I mentioned before in terms of counting more photons, et cetera, being smaller, it also is still able to resolve the expected absorption and scattering coefficients. And so in figure 15 on the left side, what you see are these, what are actually scatter plots of measurements taken each in a 5 millisecond window. So we fire the laser in these 100 picosecond pulses at a 20 megahertz frequency. So in a period of 5 milliseconds, I think we fire the laser 100,000 times, if I remember correctly off the top of my head. So In 100,000 laser pulses, we use that to get one measurement. And from that 100,000 laser pulse measurement, we're able to see what the optical absorption and scattering properties are for that phantom. And what we see in this figure on the left side is that there's pretty good clustering of our measurements around the same absorption and scattering property. There is a slight deviation from the absolute expected value, but some of that can be explained by some of the errors that occur both in calibrating and the assumptions that go into calculating the measured properties. So what we like to see is that they're tightly clustered. That means there's some amount of precision to this measurement, and we're able to reproduce it in every 5 millisecond window that we record from. The other positive result is that there's a good linear relationship between the measured expected values for both absorption and scattering. So that's what you see on the right-hand side. So you expect as the absorption scales up, you get a similar change in your measured value, and we do see that. So the deltas are good, the relative changes are good. We just have a little bit of absolute error in where the absorption and scattering properties land. And then if you kind of go ahead to the next figure, figure 16, we repeated one of the stability tests that we did early on. Well, it's actually a little different. So early on, 10 minutes ago, I was talking about the IRF stability, the stability of our system and how well it performs over time. And here, what we're looking at, we have a measure of a known optical phantom, and we're looking at how accurate we're able to measure that both the absorption and scattering properties over time. And so we plot of measured absorption over a 2-hour window. So we collected the data for 2 hours straight, and you can see the estimated absorption for both of the wavelengths we use, which are 690 nanometers and 850 nanometers. And the shaded regions show plus or minus 5%, so we're well within 5% over the whole time period. All right, so that kind of wraps up the stuff I wanted to highlight on the phantom data side. All of this is great, and it shows the characteristics of the system and that it's working as we expect it, and it kind of matches with the fundamentals of the time domain fNIRS technique. However, what we really care about is recording signals from the brain of a human. And while the paper mostly focused on the phantom work, I want to switch gears a little bit and talk about what's at the end, which are the human measurement results. So the first kind of important thing to look at is in figure 17, you can see this nice signal that shows what looks like if you had asked me to draw like the waves in the ocean or something. What this is a heart rate signal from a person that's wearing the Flow device. And the reason that I want to point this out is this is extremely rare for a time domain system to be able to domain systems integrate for about a 1-second measurement. So where I mentioned before, we're using 100,000 laser pulses spanning a time period of about 5 milliseconds. Most fNIRS, time domain fNIRS systems measure for a whole second. And because they're measuring for a whole second for every data point, they're not able to see a very nice, clean heart rate like this. And I should think it's really important that I point this out, because one of our biggest challenges is dealing with physiological signals that we measure that are not brain data or not brain signals. And so this is what would be called biological noise. It's everything else that's going on in the body, your heart rate, your respiration rate, your changes in blood pressure. All of these biological noise signals can be much larger than the actual neural signal or brain signals that you're trying to measure. So by having the ability to precisely characterize things like the heart rate, it makes it easier to remove it from the data that you measure, so that you can look at just the neural responses, the brain activity. So I'll kind of skip ahead now to figure 19, which is the real functional brain imaging task. So here what we've done is a kind of classical motor task where we had two different participants here. One of them did 2 sessions, one did a single session of finger tapping. And so what the task is each person is prompted to either tap their fingers together on their left hand or their right hand based on a cue. And it's well known and understood that when you use your right hand, the motor cortex on the left side of your brain will activate. And when you use your left hand, the right side motor cortex activates. So this is a commonly used experiment because we know what to expect. We know that when the left hand's tapping, we want to see activation on the right side. So in the figure, in the kind of center of the figure, there's this cartoon head picture. And we show the results for left minus right tapping. So this is when a person taps their left hand, what the brain activation looks like, minus what the brain activation looks like from when they tap their right hand. And you see these two localized areas on the left. There's a blue patch, which corresponds to a deactivation, as we expect. So when they're tapping their left hand, the left side of their brain is less active. And on the right, you see this red patch, which it corresponds to an increase in activation. So when you tap your left hand, the right side is more active. So this is expected and everything looks great. And if you look at the waveforms above, you can see the actual signals that were collected during one of these experiments. And the kind of pink or reddish color bars or regions denote when the cue, the prompt, told them to tap their right hand, and the green bars represent when the prompt told them to tap their left hand. And these waveforms are from a location on the right motor cortex. So what you would expect is an increase in blood oxygenation during the green regions, which you can clearly see in the waveforms there. And then similarly, you kind of expect the reverse when the right hand is tapping. So with all this, it's been about 30 minutes now. I'll just conclude and say that what I really wanted to highlight is that we've taken this room-sized technology and miniaturized it so that it can be head-worn. And not only have we done that miniaturization, we've also managed to exceed or maintain the performance of systems that were previously state of the art. So I'll just stop there and thank you for taking the time to listen to that and to kind of look at the work that everyone at Kernel has been doing. And I'm happy to answer any questions that you all may have.
Thank you so much, Ryan, for this really great guiding us through this really wonderful work. It's so impressive, but I'll give everyone here a chance to speak. So go ahead, flash your mics or go in PTR. Yeah, hey, Serena, go ahead.
Fascinating work. Really, it looks, like you've just really nailed this thing. High resolution, temporal resolution. Is it the case in the takeaway from Figure 18 that you can go, you're getting measurements 6 centimeters in? And
Okay. Yeah. So figure 18 is not into the brain, it's across the brain. So that is how much signal we measure from any one laser on the headset to any one detector. And so we have the whole head covered. So there's a range of spacings from, say, one laser will have some detectors that are 10 millimeters away, some that are 20, some that are as far out as 60 millimeters. But what you can really see from that figure is that at about 35 millimeters away from the laser is where the signal kind of flattens out. So we're not really seeing any measurement beyond 35 millimeters on the head.
okay, no, just fascinating work. I'm going to give other people a chance to ask questions, but I certainly have more as we go.
Thank you.
Hi, Ryan. This is Dennis. I had a quick question. It's probably out of scope, but I was wondering if there were any people that you had an opportunity to test this tech with who had COVID, or if you can see any sort of application for this technology to help diagnose or treat people who are suffering with long COVID right now.
Yeah, that's a really great question. We have not measured anyone with COVID. So all of our experiments and studies are done under IRB review. And so all of our protocols specifically have COVID testing and vaccination requirements to avoid exposing kernel employees who are running the experiments from potential COVID. So we haven't done that. In terms of diagnosing COVID, I think it would probably just be easier to use a more traditional blood oxygenation monitor than brain blood oxygenation monitor. Long COVID is something that we're interested in and have thought about. And if there was a group or organization that wanted to sponsor some research in that domain, we would be in support of, because we know that there's a lot of I don't know, anxiety, stress, cognitive issues that have been associated with COVID. And that is something that you could measure with a device like ours.
Thank you for your answer.
You're welcome.
Hi, Ryan. Thanks for the very detailed explanation of your excellent paper there. And I have two questions. The first of all, in your setup, using the Apple Watch example, which I saw their flyers, they use some sort of there's an angle, oblique angle, to fire the laser and take the signal. So in your case, you have the sample, your light actually go through the sample, right? Or
Yeah, that's a good question. So we work in what's typically called reflectance mode, but the light is not actually reflecting off of the skin. What happens is we fire the laser directly into the head, and you think of a laser pulse and just think of it as injecting something like 10 billion photons all at once into the head. And so if we look at kind of like the particle theory of light, these photons are going to bounce around as they scatter off of cells in the head, whether that is in the scalp, the skull, or the brain. And so what you end up seeing is like this, it's a diffusion process really, where we inject all these photons at one point, and light diffuses kind of horizontally, and very small amounts of it are picked up at the detector side. So for every, 10 billion photons we put in, maybe we get, 50 or 100 photons that we can measure at the detector. And that's why we, pulse 100,000 laser pulses per measurement, because with 100,000 laser pulses, we can build up a measurement of tens of millions of photons, and that gives us pretty good accuracy of the measure. And just kind of remind everyone too, the fundamental principle is the same as if you take the flashlight of your cell phone and put it on the palm of your hand. You can see that you don't have just that spot of the flashlight where the light is emitted. The entire hand kind of has this glow to it, and that's because of the same kind of diffusion process. You can see this in action, where if you put light in at a point, it actually spreads out quite a lot through the tissue due to all the scattering.
I see. Thanks. And my second question is the shrink. I understand this is a collaborative work, but can you comment on what is the bottleneck that you overcome enable you to shrink down from? The earlier code you had. The previous, bulky and heavy. Is the laser module or detector or the chip, or of course going CMOS probably is one winning strategy, right?
Yeah, so it's all of the above. So Kernel really is a full stack company. We have people that work at every level from kind of like the single photon all the way up to machine learning. And so I actually started working on, my PhD was in a similar technique doing time correlated single photon counting. So, pulsing lasers of light and detecting these single photons as they get detected. And, that's kind of at the device level where we started with the integration is we focused on the detector and the detector electronics and how we could miniaturize and integrate as much of those as possible. However, if you look at the first laser we bought, was probably twice the size of a typical microwave. It was a big, bulky device, and it could do so many things. It had so many options, and all these different knobs you can turn for it. And what we did is we just pulled out what was essential and designed our own laser drivers and laser circuitry where we could use off-the-shelf laser diodes, but drive them in a way that allows us to generate these really short pulses of laser light. And that's as important as the detector design. And then everything up the stack from there, the microcontrollers that read the data, the signal processing that cleans it, the analysis that's done, even the way that the neuroscience is structured, the way we generate and present the stimulus to see what the brain reacts to it. All of that is kind of done by Kernel in a very full stack manner.
Thank you.
You're
I have a
welcome.
question, Ryan. Is it possible to sacrifice like a temporal signal if you did something like a two-photon approach, like with a longer wavelength? I imagine because you're talking about very few photons out, that maybe the signal would be very slow. But is there a way to apply something like that in a kind of compact format?
Yeah. So two photon microscopy is really interesting because what you're effectively doing is combining the energy of two photons that arrive at a fluorophore or some kind of fluorescent molecule at the same time temporally. So you kind of have two photons at the same place, same time, that give you twice as much energy and allow you to excite it. And that gives you that better depth penetration to kind of get down and focus where you want. This is a little different in that we actually get our photons spread out over many nanoseconds because each photon that goes into the brain is scattering around through the tissue before it comes back out. And we choose the wavelengths of light, 690 and 850, based on the properties of hemoglobin and its like two states, oxygenated versus deoxygenated. We could go to longer wavelengths. We go to 1064 instead of 850, but the downside to that is that you then need an exotic detector again in order to measure those long wavelengths because the band gap of silicon is such that you can't measure anything beyond a 1100 micron or 1100 nanometers about.
Thanks.
So, yeah, sure.
Curious about this spatial resolution you can derive in these dense arrays. How certain can you know where the signal came from?
Yeah. Thanks for asking the $1 million question. This is the one that that everyone brings up, and it's a really, really difficult question to answer because it's hard to get ground truth, right? Like it's hard to know exactly how big of a volume was activated in the brain, and to what extent, how much of an absorption change was that. So our best proxy is to look at these phantom experiments, which are things like the NeurOpt protocol, where we have a known device and we look at our ability to resolve it. So I don't have a specific example or number to give you, but I will say that TD-fNIRS devices typically have come up with clever experiments to show that they're able to get in the range of 1 to 2 centimeters resolution within the brain. So it's not the same resolution as you would get from an fMRI, But it's, within an order of magnitude. And, when you kind of weigh some of the benefits of being portable and lower cost and just generally more accessible, maybe that's an acceptable trade-off for you.
Well, I wonder if, knowing the mapping between function and regions, if you can build up a spatial map of registration through different types of experiments, having the subject do different things with different parts of their body or different types of language areas or other types of signals, and derive a registration that's patient specific, but then allows you to characterize more spatial resolution.
Yeah, so if you do have access to a scanner, an MRI scanner, and can do fMRI with the same paradigm on the same subject and do co-registration between the fNIRS and the fMRI, then you can compare if how close does the fNIRS data get to the fMRI data in terms of its spatial localization. That's not something we've done, but it is something that can be done. And people tend to look at things like retinotopy as a very common one, where you're looking at kind of the projection of the visual field onto the the back of the cortex, the occipital cortex.
Well, can you comment on some of your near-term use cases now that you've achieved this level of readiness?
Sure. Yeah, we've had quite a few public announcements recently. So one is we're working with a psychedelic company to do a proof of concept study on wearing Kernel Flow while participants, we're doing healthy controls in this case, or healthy participants um, undergo a psychedelic experiment using ketamine. So we'll measure their brain responses while they undergo a ketamine treatment, as is typically done for some treatments of depression or other things. So that's one kind of proof of concept that we're in progress on, and I think the first session is scheduled to happen in the next week. The other, we have this gaming study that we did with a company that's actually based out of New York called State Space. They spun out of NYU, I think, and they make a first-person shooter training game called Aim Lab, and one of the kind of training tasks in it is called Grid Shot. And so we did a little demo with a professional gamer named Scump, who is a world champion Call of Duty player. And we measured his brain while he was doing this training program, and we measured our CEO's brain while he was doing it, not being a professional gamer. And so what we found is that there are differences between a novice and a professional gamer's brain. And then we kind of recruited people in to do the experiment more and see, if we could build up some group statistics to show what those differences are, and which of those are statistically significant. So that's our gaming study. We're in the process of starting up we also have an ongoing attention study, which looks at meditation and its effect on mindfulness or mind wandering. So being able to measure changes in focus state or mind wandering state. We're about to start an emotion classification study. So can we measure signatures of the brain that become apparent when a person either views a emotional video or listens to an emotional soundtrack. And here again, we're using labeled clips, whether video or audio, to kind of have this ground truth of what is what the expected emotion is. So we can use that to build a classifier and use brain data to classify emotional state. And then the third one that we're just about to start piloting is around measuring healthy aging. So we're looking at, I think, 11 different brain-based markers that we measure with Kernel Flow, and how they change over the entire population's age range. So we're looking at individuals from 18 to 75 and how certain markers change based on age group. And what we're looking for there is a kind of trajectory of normal aging. What does healthy aging look like as you progress through the course of your life? And we think this is significant because a lot of future interventions, specifically targeted at advanced aging, cognitive decline, dementia, etc. They may be more effective if you can have a regular test to find out where someone's brain function is versus where it's expected to be based on their age. So some kind of early warning. And yeah, there's one more partnership we're just starting up that I don't want to talk too much about yet, but there's other stuff in the pipeline, and we're really trying to broaden out the applications and areas that we're doing work on. So all those things I just described to you are being done by Kernel through IRB approved studies, and working to really demonstrate some of the applications to using Kernel Flow.
Yeah, I wanted to ask, so what is more or less the estimated cost of the device? And do you think, because you talked about using it maybe for diagnosis or pre-diagnosis, you think it's broad, like it's in the range that like primary care doctors could just, and people starting in their 50s or so, screening for Parkinson, or like use it in general, large population for screening for neurodegenerative diseases?
Yeah, that's a great question. So right now, our devices, we're currently in low production volume. So we're producing around 100 devices this year. And so our production costs put the price of the device around $100,000. As we generate some demand for our devices, then our volumes will increase and the cost will go down quite significantly. So as I mentioned early on, every decision we made about the hardware was made in order to make sure that as we start to hit volumes of 1,000 and 10,000, 100,000, or 1,000,000 devices in the field, we can start to see those costs drop down. And our estimate is that if we have very widespread adoption of this device, we can get the whole headset price eventually down under $5,000. But today it's 100,000, and long term, our objective, I think we stated a year or two ago, is to kind of make it so that everyone could have a kernel flow in their home by 2033. So kind of piggybacking on that mainframe to PC concept, most people couldn't own a mainframe. It's far too expensive and power hungry. But once PCs were available, it was still expensive. It took a lot. I remember when I got my first PC, it was not cheap. And I know my parents worked hard to make sure we could have one. And that's where Kernel Flow will start, and eventually we'll get to a place where it's in the range of an iPhone. But I think that's going to happen over kind of the next decade or something. So I will also add that, based on some of the work from these initial application explorations, we may find that only certain regions of coverage are needed in order to drive the measurements that are required to do each application. So if you think back to, I don't know, like I mentioned the mindfulness or attention task, a lot of the brain that's involved in your attention is all in the prefrontal cortex. So you may not need a whole head coverage system. You may just need about 10% of the system that only covers the prefrontal cortex. So another option for reducing costs long term and making it accessible to everyone is to just tailor the coverage to the application that's needed.
Another question, since we are on Clubhouse and there are a lot of transhumanists, singularity, all kinds of people here on Clubhouse, I was thinking, so could is the data precise enough that you could basically over your lifetime record brain activity, and once you lose basically your own brain capacities, that you have collected enough data to basically uh, train a chip or some AI to take over what usually your brain would do? Do you think that's also something? Are people approaching you from this sort of world, basically?
uh, no, not exactly. We haven't been approached by anyone that's looking to kind of upload their brain. Um, so I think that I'll qualify this by saying I'm not a neuroscientist. I'm an electrical engineer and a physicist at heart. I've worked at a neurotech company for almost 4 years now, so I've got a kind of understanding, but by no means am I an expert in this field. Um, and what I would say in response to the question is that with fNIRS specifically, and in this non-invasive form that it's currently in, we're limited to measuring just the outer layer of the brain, really. So there's a lot of brain that we're not able to access. And the reason that this is effective as a tool for understanding things like cognition or pain or anxiety or stress is that your cortex is involved in a lot of these functions, even if it's not the only thing involved. So you may have deeper structures in the brain that are handling some of the processes associated with whatever the experience is, and only a small portion of it is reflected in the cortex. So what I would say is that you would have very incomplete information about the details of what you were experiencing. But perhaps you could have a recording of certain emotional responses to things. So, what your likes or dislikes are, or, how you felt when you saw that person you really cared about, your parents, your child, your spouse or love. Those types of things I think could be recorded and kept, and you would have some indication of kind of these brain states that are represented in the cortex. But I don't think the technology we've built today is the answer to uploading all your memories and deepest secrets into the cloud.
So I'm sure you're looking at deriving control surfaces. I'm curious what you can say about that at this point, and whether there's reliable control that the subject can exert that's detectable that could derive external devices.
Yeah, so this is a very common question too, and I kind of gave a little preface at the beginning of the talk that the technology we've built is around hemodynamic responses. So everything we measure, we're measuring a lagging signal of what's actually happening in the brain. So when your neurons start to fire, the cardiovascular system responds by providing this oxygenated blood to use. So I personally don't think fNIRS is a great solution for control type of applications because of this lag, right? Anytime you're basing things on changes in blood oxygenation, you're going to lag the neuronal activity. That said, there has been some work done on what people call a fast optical signal. This is not something we've explored at Kernel, but the theory behind it is that it's also called an event-related optical signal. And Gratton's group is the one that has done a lot of work on this. And the idea is that when neurons start to fire, there's a localized change in scattering that happens much faster than the change in absorption. And you may remember that our system measures both changes in absorption and scattering. However, we don't believe we have the SNR to measure individual changes, like event-related changes, single trial changes due to scattering. So again, I think it's a little bit of a reach for like a good control application. And so instead we focus a lot on measurement and kind of understanding of the mind, right? Really a better understanding of oneself rather than as a control mechanism.
Can I
Yeah, I have a couple
oh, go
questions.
ahead. Go.
Hi, Ryan. Thanks for spending time on with this and with us answering questions. Um, so I'm sorry, I have to admit I'm a little unfamiliar with the specifics of the device. Um, so I'm curious about what the spatial and temporal resolution um, looks like, and also I'm curious because you were talking about tracking hemoglobin, if it has the same kind of bottlenecks that we have with fMRI or anything that's tracking um, blood oxygen, blood flow, metabolism kind of things, in terms of being able to relate it to the firing. Obviously there's a latency, but in terms of all the different potential mechanisms that could be driving a blood flow or blood oxygen signal, um, or if this technique offers any advantages into relating it to the underlying neural activity.
Yeah, so it is similar in signal to fMRI in that you're measuring hemodynamics and kind of that type of rate of change. Our temporal resolution at an individual detector level is 200 hertz or 5 milliseconds. However, we do a laser pattern to measure across the entire head without crosstalk. And that means we image the whole head at between 7 and 8 hertz right now. So we're getting a full image of the head every, I don't know, 100 and something milliseconds or so. So that's the type of temporal resolution we're talking about. We're not talking about millisecond type of resolution across the whole head. And if you were to ask me what our main value proposition is, it's a signal like fMRI, but in a portable, compact, low-cost form that's easy to operate. And the trade-off you're making is you only get access to the cortex, whereas in fMRI you get the full brain.
I see. Yeah, that was my next question, was the depth, like how far can you go?
Yeah, so in the publication, we show kind of contrast out to about 20 millimeters in a phantom, 25 millimeters. The typical or average gap between scalp and brain of a head is about 15 millimeters. So we're somewhere in the neighborhood of, say, 5 to 10 millimeters into the brain.
Awesome. Yeah, thank you. I just was curious about those details. The what you're building is super interesting, and the portability is irreplaceable. That's really cool. Thanks.
Yeah, thank you. I need to go in a little bit, but I could probably answer one or two more questions if anyone has something.
Yeah, please go ahead. Whoever has a last question, and then we'll close the room.
Well, I'm if
I think you're breaking up. Try
we need
again.
to remove our hairs when we wear the device because the attenuation.
You were breaking
Sorry.
up
Oh,
shortly, but yeah, go ahead.
What about now?
I can hear you.
Yep.
Hello. Yeah. Okay. So I'm wondering, when we wear your device, do we need to remove our hairs? Because I think the hair is black or yellow or some color, it can attenuate the signal greatly. That's the question. And I also have a second question. So comparing the time domain measurement, what about the frequency domain measurement like OCT? Okay, thank you.
Okay, yeah, so two questions. Hair, yes, is a I would say, classic problem with fNIRS. It's true of any fNIRS system. If someone is working on an optical system and tells you that hair is not a problem, don't trust them. So I will tell you right now, hair is a problem. And what we've done is worked really hard on the mechanical design of our system to make the waveguides that we use to couple the light to and from the head as much like a comb as possible. And the reason we make them comb-like is so that they can work through the hair and you can get access to the scalp even if a person has hair. So we would appreciate, yeah, sure, if you want to shave your head, we'll get great signals. I promise you that. But we don't want to require that as a first step in using one of our systems. So hair is a problem. Dark hair is obviously the biggest problem. Light hair is less so. So yes. And then in terms of frequency domain, there are also frequency domain systems that have been on the market. Still, I have yet to see anything that's been miniaturized, but they use a phase delay measurement as the measurement, and you can also get both absorption and scattering properties from the frequency domain measurement. However, the big downside is that they're only able to assume bulk tissue properties. So unlike the time domain fNIRS measurement where you get this kind of full time of flight curve that shows the average time each photon has traveled through the tissue, frequency domain system only have one thing that they're measuring, which is this phase change. There are other techniques. There are some groups working on what's called inNIRS, so an interferometric version of a NIRS measurement system. The challenges with that, again, are really around miniaturization. I haven't seen anything compelling that enables that technology to be miniaturized in a kind of scalable way. So there are tons of techniques for probing the brain non-invasively, and one of the reasons that Kernel picked time domain fNIRS is because we could check all these boxes. We get as much information as possible. We can build the electronics and the optics in a way that allows us to scale it up. And we see a path to kind of a mass market system in the future. And the insight we had was we looked at these other things like frequency domain NIRS and also inNIRS, and the insight was really that what we did is time domain fNIRS, as we built it, is as close to a digital technology as possible. So from the moment we detect a photon, everything else is happening in the digital domain. Frequency domain system relies heavily on analog electronics, and so you add a lot of circuit complexity to that measure. Something like inNIRS adds a lot of optical complexity. So we tried to remove all of the complexity that we could and push it into the digital domain because we know that digital things scale well. And so our technology is built to scale around all these digital things that our world has been built on in the last decades.
Yeah, with that, I wanted, to thank you so much for giving this great presentation and for your amazing work. I think this will enable also in research so many more avenues that we can go while people, are moving around. To have precise brain imaging is quite amazing, and it's way cheaper. So I think also for research, this will be, very useful and quite a breakthrough. So yeah, again, we are very honored that you came here to our club, and we wish that millions of people order it so everyone can afford one soon. So, and that, the semiconductor shortage doesn't delay things for too long. So yeah, thank you so much. We appreciate it.
Yes, thank you for having me. Yeah, tell all your friends, everyone get one, and we'll all win. So yeah.
I'll take 2 to go.
Okay. Well, thanks, and happy to come back sometime if we have something new to talk about. I'd be happy to.
Oh yeah,
So,
that'd be
thank you all.
amazing. Yeah, please come back when you have news. Thank you so much. We'll
Thank you,
stay
Ryan.
in touch. Bye, Ryan.
Okay. Thank you. Bye.
Thank you.
Thanks, Ryan. Thanks, Katerina.
Yeah,
Thank
sure.
you.
Yeah. Thanks everyone. That was the second room today. I hope everyone enjoyed it. I just make quick announcements. Tomorrow we'll have news, like science news that are around health. I found like very interesting research that came up the last day, so I'll share some of that. Then Ryan will lead some tech innovations in his field, the room, and then next week we will have on Tuesday the guest invited speaker that will talk about his paper. It's stochastic microbiome assembly depends on context. I had a lot of people that wanted to hear from a microbiome researcher. Over time I had a lot of requests, so yeah, I'm glad that he will be coming next week, February 22nd, and then Dr. Levine will come and will talk about his technical logical approach for mind everywhere, and it's a synthetic biology and bioengineering technology that will provide us more insights into cognition and consciousness. So Dr. Levine was here before with his colleague Josh Wongert. They talked about the self-replicating xenobots, and Dr. Levine does a lot of very interesting, amazing projects. So he will present another one of his. And then we'll have Dr. Kaufer, and it's about increased myelin linked to anxiety and PTSD. So it's also a new paper that came out that will be discussed here. So yeah, come back and enjoy the rest of your evening, day, morning, wherever you are. And thanks for coming. Okay, I'll close the room now. So bye everyone. Thanks for coming. This was a great day today with interesting rooms.
Thanks to
So
Katerina.
bye. Thanks.